This is going to be the lab on paper chromatography, and we're going to read a little bit of this information in this paragraph, and then I'm going to go ahead and do a time-lapse video of the actual procedure, and then what we're going to do is then we'll have a summary of our results. So I want to point out a few things, and that's in this first paragraph. Again, chromatography, chroma means color, and graphy means to graph or to chart. So many inks are actually mixtures made up of several basic pigments. Each pigment may have a different color, a different structure, and different physical and chemical properties. The pigments in an ink mixture may be separated using paper chromatography. So for example, I'm going to be having different types of inks. I'm going to have whiteboard markers like this, okay? I'm also going to have some Sharpie markers, and then I'm also going to have some Crayola markers. They're going to be very different types of pens. And we're going to see that this purple coloration and this red coloration and this green coloration and this black and this pink, these are not truly the pure colors that are in the pen, but they're actually a mixture of various colors. So if you can write over here, we're looking at chromatography. The colors that make this purple are actually, they're dissolved in the liquid that's in the inside of this pen and at the tip of that pen. So again, with chromatography, we are separating two dissolved solids. So again, two dissolved solids. That's the first point I want to repeat from our lecture notes. And that's the first point. Second thing, paper chromatography is an example or more general type of chromatography called adsorption chromatography. Okay, if you remember, adsorption means that it's going to stick to the paper. The paper acts as an adsorbent, a solid which is capable of attracting and binding the components in a mixture. So here's a small picture down below here. Imagine this is the structure of the paper, and it has these, like a sponge, has these tunnels or holes or caverns. How much is the particle going to then stick to this material? That is the adsorption. So again, adsorption is going to be sticking more. Okay? The mixture to be separated is spotted onto a surface of a paper. Here's the paper that I'm going to be spotting it on. And you can see that I have my samples of various different types of colors. We'll get to that in a few minutes. Okay, onto the surface of the paper, and a solvent is allowed to seep or flow through the paper. If one of the components in the mixture is more strongly adsorbed onto the paper than another, it will spend a smaller fraction of the time free in solution, will move up the paper more slowly than the solvent. This is another key point. If one of the components in the mixture is more strongly adsorbed, okay, so if we increase adsorption, again, adsorption it's the sticking to. That means it's going to stick more. So if we increase absorption, it's going to stick more to the surface of the paper rather than moving up the paper itself. So if it's sticking more to the paper, that means it's going to move more slowly. Okay, so for example, we have our paper right here. If this adsorbed more to the paper than this right here, it would move very slowly, maybe out to here. But if this one has less adsorption, it's going to stick less, it's going to flow faster through the paper material. So components that are not strongly adsorbed, so in this process, so here's number 2, we're going to also decrease adsorption. If you decrease the adsorption, it's the opposite. It's not going to stick more, it's going to stick less. It sticks less to the paper. If it sticks less to the paper, That means it's going to move faster through the paper. Those are the main concepts of this lab. We're separating two solids, and one of them is by the type of material and the adsorption. So in this process, what I'm going to do is, here is my filter paper. This is the chromatography paper. And I have various substances that we're going to be seeing how quickly they move out, but also not only how quickly, but what are these colors made of? What I'm going to do is, I've already pre-assembled this cone. These are the instructions on the other side. Here's the chromatography paper. We marked it with dots. I've made this little cone out of the same material. This little cone is going to stick through the hole here, okay? And then I'm going to sit this in this container of water. So as the cone sits in the water, it's going to absorb the water up the cone, and then when it reaches the top of the cone, it's then going to reach this chromatography paper. Then it's going to spread outward in all the various directions. So I'm going to now film a time-lapse video for you guys, which is the next part for this lab, and then you can watch that, and then we'll come back and we'll discuss the end results.